Um, sure, yeah, can everyone see the slides?
Yes, looks
Okay,
good.
that's great. So thanks Alex for the very kind introduction, and it's really my great pleasure to talk again with the bioinformatics community, QCBio community. It's really my great pleasure to have probably very last chance to talk with people here. So today I'm still going to talk about some of my work in my PhD, which is trying to identify the gene regulatory strategies for immune response genes. Honestly, at the beginning, I have no idea like how this is gonna prepare my next step, my next career. But through my job searching, job hunting with all the pharmaceutical companies, particularly on the immuno-oncology side, I found actually people generally are quite interested in understanding how to efficiently to identify the drug target from the perturbation data, screening data. So that's why I feel like this work has really prepared myself for my next step. And hopefully today I also can touch a little bit on how such work or like PhD work, like basic work, can prepare ourselves to be a something useful or valuable for the pharmaceutical industries. I hope that's something I can touch today. So as you all probably know, like our lab is very interested in the innate immune cells. We are the system immunology lab. So in our lab, we are particularly interested in the macrophage, which is a immune sentinel cells that can recognize a broad spectrum of pathogens and generate the pathogen specific gene response. Then the key question is how the macrophage can sense a pathogen information and propagate information through signaling pathways to precisely control the gene expression. So the very attractive ideas such gene-specific response can be achieved by combinatorial gene regulatory strategies by multiple signal-dependent transfer factors. So here's the interferon beta, which is has been shown regulated by AP1RF and MFRB. However, it's still unclear how those transfer factors together synergistically regulate interferon beta, and is there any dominant signaling pathway regulate interferon beta gene expression, or like are they synergistically, how they synergistically regulate together? So all those questions. And moreover, what about the other immune response genes? Can we identify the gene regulatory strategies for them? So actually, I would see now when I review this topic slides, I've talked tons of times. I've really amazed by the biology system. Like it's so robust. Like if many people have so many mutations, it's still manageable to defense lots of pathogens. Like in different conditions, we are still possible to defense all those pathogens. Certainly the redundant design between different signaling pathways is certainly very evolutionary results of all the biology systems, and which is great, but it also causes lots of problems when we're trying to fully understand them, since there are so many redundancies between signaling pathways. And it actually turns out to be the I think one of the biggest challenge in understanding the gene regulatory strategies in this field. So actually, you can see understanding such transcription control of immune response genes actually it's not actually limited on the infectious disease. It's actually broadly recognized as one of the central questions in immunology. So this usually can lead to discoveries of the novel drug targets. So here I'm going to talk about, briefly mention two very broad field in immuno-oncology side. One is study MHC class 1 gene. So this gene, this molecular has been found well correlated with tumor resistance, resistance to the PD-1, PD-L1 treatment, as that if you lose this MHC class 1 molecular, you lose the ability to present antigen. So the T cell cannot recognize the tumor cells, and therefore you cannot really kill tumors. So the key idea is if we can find the potential regulators that can activate this MHC class 1 gene expression, then we can potentially recover the antigen presentation, therefore can enhance the anti-tumor immunity. So another example is from FOXP3 in regulatory T cells. So Treg cell, actually the development and function are dependent on the FOXP3, and we sort of know the the regulatory T cells play a immune suppressive role in tumor microenvironment. So therefore, if we can identify the potential regulators that can potentially reduce the FOXP3 gene expression, then we can possibly reduce the Treg cell functions, therefore enhance anti-tumor immunity. So actually in this field, people are nowadays really actively use CRISPR screening approach, trying to identify all the potential drug targets and gene regulatory strategies for all those immune checkpoints molecular, like PD-L1, CD47, interferon gamma, and TNF. And what is more exciting recently, I just found that people use combinatorial CRISPR screening approach to get more information about the redundancy between different regulators from Jonathan Weissman lab, and which is really exciting. I think it's going to be very promising to fully understand the gene regulatory strategies. However, you probably can see the purely experiment approach are insufficient to identify the gene regulatory strategies. So let's see for the RNA-seq, which can capture the functional interactions, we usually have false positive and negatives. That's due to the indirect secondary effects and incomplete knockdown and compensation. I would say compensation is a big issue here. And also for the ChIP-seq, it's also have lots of false positives and negatives. That's due to many peaks may not be functional role, playing a functional role, or maybe distal binding events. So there is still a great challenge that people trying to link those enhancer with the promoter with the gene interaction. This is also a big challenge in the gene regulatory study field. So then the key idea is how we can integrate RNA-seq from multiple perturbation conditions within the same computational framework. And then we probably want to fully understand gene regulatory strategies, mostly from perturbation data rather than from the functional data captured by the ChIP-seq. And therefore, I've developed the computational framework trying to gain quantitative and mechanistic insights for combinatorial gene regulation. So before I move forward, you must wonder, like, is this a really necessary to develop new computational framework, or what are the limitations for the traditional approach to identify the gene regulatory strategies? So I think the traditional approach is mostly generate a bunch of knockout, either by CRISPR or shRNA, so that you can directly compare the gene expression with in the knockout with the wild type. Then you can identify a group of genes that has shown reduction. Therefore, let's see, for this group of gene, this may depend on this regulator. And if you do the same thing for another regulator, then you can identify another group of gene. And then people overlap them together and recognize those overlapped group of gene would be the gene dependent on the both regulators, which is great. However, this approach, simple approach, sometimes cannot handle situations that when TFs are not singularly activated or completely knocked down with different ligand stimulation. Let's see here, if we have for wild type multiple transfactor activated, and if we've got knockout in mediate, none of them are fully diminished. And secondly, is the big issue is compensation between transfactors. Like, let's see, if you knock, there's one gene, like let's call gene X, depends on transfactor A and transcription factor B, and both can activate this gene expression. And if we simply knock down transcription factor A, we still can get gene expression. But actually, this not because this gene not depend on this transfer factor, but actually because there's a potential functional redundancy or compensation between those two transfer factors. And this turns out to be the key that you cannot use this simple approach to fully dissect or understand the potential regulation roles behind of all those immune response genes and all the other genes. And also, it's hard to handle time course RNA-seq data and hard to handle combinations of multiple transfer factor dynamics. Let's here you have dynamics of that rather than the simple up and down, then how you dissect of that, which is all limitations of traditional simple approach. So therefore, for us, we have generating a bunch of perturbation data for the bone marrow-derived macrophage, and we are asking what if we can measure TF activities as input and nascent RNA or RNA expression as output, whether we can use a computational framework and trying to dissect such gene-regulated strategies. And after we identify of that, whether we can use additional functional genomics assay to validate or fully understand the regulatory mechanisms, let's say ATAC-seq and TF chip-seq. So therefore, for my research, the key goal is trying to identify the gene-regulated strategies for individual genes, since this is more meaningful in terms of more practically for the drug target development. Or fully understand mechanistic insights. And also secondly is how we can rigorously determine GRS by considering experimental data uncertainty. And this is all well-known problems, like you have multiple data uncertainties, then how you can have really rigorous determinants of that. And finally, how we can gain mechanistic insights on immune response genes. So those aims actually turns out to be really challenging. We actually don't know how to reliably and accurately measure data uncertainty for time course RNA-seq data, and we are not sure how should a computational framework be designed to efficiently leverage all available data sets with various data quality at the beginning. Now we understand more, and how to gain mechanistic insights on immune response gene regulation. And finally, the solution would be develop a novel statistical error model to quantify time course RNA-seq data. And secondly, we developed a Bayesian framework to infer GRS from multiple data sets with various qualities. And finally, we examine the chip-seq and ATAC-seq to get mechanistic insights about those gene regulatory strategies. So this work collaborated with Smil Lab and Anoop, which is postdoc in our lab. So we have focused on the bone marrow macrophage, which is a primary cell, and we generate a bunch of genetic knockouts and use different ligand stimulations trying to perturb the bone marrow macrophage. And we have measured time course RNA-seq data. And here we mostly focus on 3 primary immune response signaling pathways and their downstream transfer factors, which is FKB, IRF, and MAPK. So we have measured TF activities by epsilon and Western blotting, and also nascent RNA expression by chromatin-associated RNA-seq. And we have quantified their gene expression here, nascent RNA expression by chromatin-associated RNA-seq. And also we have measured or quantified TF activities in nucleus with epsilon and Western blotting. So, so for the macrophage here, after the ligand stimulations, the macrophage will sense of ligand information, then propagate such information through signaling pathways, then translocate the transfer factor from cytoplasm to nucleus. And for those transcription factors in nucleus, it will bind into either enhancer or promoter, and then recruit the polymerase II and initiate transcription. And such process actually can be captured by the ODE or mechanistic models we have developed here. And particularly for the promoter activity can be captured by this FT. And for the combinatorial gene regulated strategies actually can be captured by this GRS, gene regulated strategies. And this function actually derived from thermodynamic models, which can be really useful to capture different logic gate between transfer factors. And for the regulation strength of each transfer factor, we use Hill function here to derive, to describe their regulation strengths. So at the beginning, we actually focus on the very synergistic and non-synergistic regulation, which is a very simple case. And we have we have used the AND gate or synergistic term to describe that only two TFs together can activate this gene expression. And for the non-synergistic or OR gate is that any of the transfer factor can activate this gene expression. And here is the logic gate derived from thermodynamic model to describe the the potential logic gates. And here we have focused on three transfer factors and enumerate all possible AND and OR gate between each two transfer factors. And then we have exhaustively enumerate all possible logic gates between three transfer factors. You can see single, dual, and triple logic gates. So therefore, with this developed the mechanistic models, we can capture the logic gates and both the TF regulation strengths. And you can see with this developed model, we can simulate gene expression. So actually, since we are working on the time course RNA-seq data, we found actually when gene sharply increased or decreased, there is huge or much higher error bars than they reach to the plateau. So what we found actually this increased error is actually due to the temporal uncertainties that caused by the sample handling or the heterogeneous cell response. Then Alex, a couple of years ago, asked me, can I really accurately estimate this? So at the beginning, I feel like this really challenging tasks. I have no idea how to do that. But I actually eventually worked with Jessica Lee, and we come up with this novel statistical models, error models that can decompose the observed data uncertainty into the volume part and temporal part. And then this error model can avoid overestimation and underestimation, which is very nice. Another big challenging for this error model is that how we can reliably estimate data uncertainty from very small sample size case. In our cases, we only have 2 replicates. You can see it's really challenging to estimate variance just with 2 replicates. So again, work with Jessica, we use we come up with this idea actually to putting all the gene or data points together that to stabilize our estimations. And we have using Bayesian rule that can combine such pooling information, which is global trend from all genes with single points so that we can generate stabilized point-specific estimation, which is here. And this approach turns out to be broadly used by RNA-seq field, which is DSeq2, Agile, and Thor. They all use it. So here is the results. We have using the simulated data to benchmark of it. So you can see this ground truth variance and this estimate variance. And we can see the pooling approach can increase the Pearson correlation from 0.46 to 0.55. And if we use time volume error model, it can increase further from 0.55 to 0.96. So after we got this error model, we also request a computational framework allow us to plug in such input-output data together with data uncertainty. So again, collaborated or worked with Jessica Lee and Sriram. I've actually come up with this Bayesian optimization framework that allow us to identify all the possible gene regulatory strategies from the data we have collected. And search all the parameters in the full parameter space. And here's again your simulate data to benchmark of these pipelines. And you can see the data from fit model can well capture of the ground truth. And if we use the compare the time value to the other things, we can see the time value model can identify all 93 gene regulatory, the logic gates. And for the time value model, it can well constrain of the parameters within twofold range. Okay, so after we got this computational framework, then the next question is, how can we use this computational framework to fully understand the gene regulatory strategies for experiment data rather than the simulated data? So this data sets we have, I've talked before, there are a couple of points that I want to emphasize still. That is, we only focus on the highly induced gene, which shows tenfold induction, so that we have strong phenotype. I think this is very, very important ideas. Like if you look at the CRISPR screening approach, lots of CRISPR actually just examining the basal level gene expression for those screening approach, which is very nice. But usually you have lots of noise and noise so that you could not identify the signals. But if you have ligand stimulations, perturbations, and combine with the CRISPR screening, and you just focus on those highly induced genes or signals, you usually will see very nice biology behind of that. And secondly, we focus on the primary immune response, which is the first order, so that we can avoid signaling cascade and feedbacks. And I think this is also very important to use the ligand stimulation, again, focus on the primary immune response rather than the later point. And also we have focused on the transcription initiation, which is TF activity and RNA expression, so that we can avoid upstream and downstream regulations from signaling crosstalk and post-transcription modifications, which is very nice. In this case, you can get a really clean signal. But I would see usually you also lose some of the other regulatory mechanisms of that. But I guess those things maybe can use different assay to identify. So therefore, we have applied the computational framework we have developed into the data we have collected, and this workflow. Again, we have the gene expression data from chromatin associated RNA-seq, and we have TF activities, and we have quantified the data uncertainty for the chromatin associated RNA-seq, and we send them together into the model fitting, and then we use these best likelihood to select or screening all possible logic gates. And then we map them back to the 17 logics. And then we can select those fitted GRS or not fitted GRS. This workflow we have developed. And then with this workflow, we have seen that very small group of genes are identified regulated by single TFs. Actually, majority of them are regulated by the 2TF and 3TFs. I think this is really shocking compared to the previous people thinking on this. But I think it also makes sense as if you have redundancy and if you don't have ligand stimulation, maybe it's hard to identify all those compensated regulation for those immune response genes. Next, we actually fully focus on the MFKB and IRF synergistic regulated genes. We have actually included all those genes and plot them here by comparing their wild-type gene expression and IRF3 knockout. And you can see we have identified all the previously reported genes, 5 genes, synergistically regulated by MFKB and IRF, showing with the black circle. And we have identified all the additional genes that are potentially compensated by MAPK and for the loss of IRF3. You can see lots of them are here. And of course, if you couldn't see the gene reduction in IRF3 knockout, you cannot select those genes as F3 regulated genes. So that's exactly the problem for the traditional approach. They cannot identify those group of genes. So after we, so here is the zooming of some of those genes we have identified. And you can see our approach allows us to identify all possible gene regulated strategies that can be explained by the data we have collected. And those are all detected as synergistically by the NF-κB and IRFs. And we further examine the ChIP-seq signal for both of the genes and both of the regulators, IRF3 and RLA from NF-κB. And you can see there's induced peak in either promoter region or intron regions, which is very nice, sort of confirm our analysis that we have detect those gene regulated strategies. Yeah, I think that's all the pretty much what I have done in my past few years. Next, I want to thank all the people who helped me, my advisor, Alex, my collaborator and committee member, Steve, and all the committee member and faculty members, Jason, Roy, Jessica, Shriram, Eric, and also collaborator, Anup, sort of Catherine a little bit, and also my mentor, Dan, and also thank all the people and community for bioinformatics program and QCBio, which is really nice community. I've got a bunch of good friends here, and I really like it, and I hope all the people in the community can can be in touch with each other more, and I hope I also can be in touch with you, and hopefully to see more great science from you, yeah, in the future, yeah. And finally, I want to thank you for listening to me again in this situation, yeah.